Alrighty, hello there, Biology 30s. So I had a couple students join me today. Well, more than a couple. We had a really good turnout today on our weekly meet for the week of, it's May the 4th. May the 4th be with you. So we went through last week's genetics formative, and one thing I needed to do was kind of clarify some terminology for students. So talking a little bit about some of the basics genetics terminology, like the fact that you've got about 10 trillion cells. Each cell has a nucleus, and inside of the nucleus, of course, is your genetic material. And our genetic material looks like these condensed chromosomes. It's X-shaped stuff. And that is where your DNA is, right? Your DNA has segments on it that are called genes. And so these DNA letters, which you'll learn about next week, code for a particular protein. And so you may have a dominant allele or a recessive allele. Maybe it codes for your eye color or your hair color. So the gene is the segment of DNA that codes for an allele, and you have two different copies of an allele. You receive maybe a dominant and a recessive from your parents. Maybe you get a dominant from dad and a recessive from mom. And that would make your genotype heterozygous. If you got two dominant alleles from your parents, then you'd be homozygous dominant. So the allele is the two different forms that the gene can take. Either it's going to be dominant, which we use a capital letter like big B, or it's going to be recessive. So a lot of times I write that as big B is dominant to little b. And then the pairing of that. So remember, you get one allele from mom, and you get one allele from dad, right? And that happens right at the time of fertilization. The sperm and the egg come together. And if you got two dominant alleles, your genotype, your combination of alleles, is homozygous dominant. And your phenotype, your physical appearance, what you look like, is going to have brown eyes. Now, as long as you have one big B, you get the brown eyes, right? Because it's dominant. It doesn't matter what that second allele is. The dominant one overpowers it and overshadows it. It masks it. So you can be heterozygous. You can carry the little b allele, but you don't show blue eyes. The only people that have blue eyes are the ones who are homozygous recessive. They have to have two little b's, two of the recessive alleles, in order to show or express the blue eyes trait. So phenotypes, physical characteristics, genotypes is the combination of alleles, and the alleles are what's coded for you on your DNA that you receive from your parents. So again, you get two letters for a genotype. And some students were writing here, what is the man? Well, he's a little u, and the woman is a big lu. No, that's not a genotype. That's just an allele. You have to pair them up together. And if I'm telling you use U's, then don't give me R's or T's, right? Use the legend that I have. So the man is little u. He's homozygous recessive. He has two of the recessive alleles. The woman, though, it says right here, is heterozygous. She has separate eyebrows. So she has a big U, and then she has a little u. Why is that? Well, because this big U is dominant to the recessive. So I'm going to post these two videos. The Amoeba sisters go through alleles and genes and Punnett square and sex-linked traits. And I'm going to harp on you guys. I'm just going to keep reminding you, please read instructions. Now, a lot of you were lucky that this time you lost marks. Sorry, you didn't lose marks because it was just a formative. But if you are not reading instructions on how it is to express your answer, or you're not reading the question carefully, then when it becomes a summative, it will affect you, right? And it will cost you. So read carefully. So here's an example. Circle the offspring that has the unibrow genotype. So a lot of students, they did the entire Punnett square correctly, but then they didn't circle unibrow, and they only got 0.5. Okay, and so some of you did that on your computer. That was perfectly fine. You highlighted or bolded those individuals. Okay, and again, this, I know we don't have a diploma this year, but in previous years when we did, if you're asked to write an answer to 2 decimal places, this student has done only 1 decimal place. That's wrong. They are not going to give you a correct mark for that. Okay, so you must read carefully. Decimal to 2 decimal places, 1 2 decimal places. All right, so please read carefully. Here's another reading error that students did. This is in the Kermode bears, the spirit bears, and it was asking for two separate things. So read carefully. It's looking for, is it male, and what is the probability of having a white coat? So a lot of you did the Punnett correctly, and you found, well, it is 1/4, and so it'd be 0.25. But you forgot they're looking for the fact this is an and, so you have to multiply the two probabilities. Probability of male and probability of white. A half times a quarter is 1/8. And then read carefully. This value is going to be rounded to 2 decimal places. So if you wrote 0.125, you did not get the answer correct. That's 3 decimal places, not 2. You had to round this, and the answer is 0.13. Okay, so reading carefully. How do I express my answer? Am I following the instructions? Am I just looking for white coats, or am I looking for a male or a female and white coat? Now, there seemed to be a fair amount of confusion also about the test cross questions. That was 3, 4, 5, 6. This was the pigeons. And so the bar pigeon, this pattern on his wings, is because he has what we call the dominant trait. So we know that this pigeon has a big B and something, because he has the dominant allele, so he has the dominant trait. This pigeon, on the other hand, has no bar back there, so this is the recessive. We know 100% by looking at her that she's little b, because she has the recessive. She has no dominant gene. She cannot have the dominant trait or phenotype. So anytime you are doing a test cross, we know this male is dominant. We know he's got the bar, but we don't know what that second allele is. Is it big B, big B? Is he homozygous dominant, or is he big B, little b? Is he heterozygous? So what phenotype of female would we use? We know the one we know. Now, this is not a phenotype. This is a genotype. So a lot of students were telling homozygous dominant or homozygous. That's not a phenotype. A phenotype would be barless, okay, or barred. It's what is the appearance? What do they look like? The physical characteristic. So in a test cross, you take the dominant, that's the bar, the male, and you mate it with the one that you absolutely know, the recessive. And you end up then trying to figure out, based on the offspring, is that male going to be homozygous dominant, or is he heterozygous? You can't tell by looking at someone with the dominant trait. You don't know what genes, what alleles they're carrying. You know what they look like, but you don't know their genotype. So the whole point of a test cross is to determine this male's actual genotype. So only 2 correct answers here. So again, a lot of students did not read that you must choose both. So here's an example of a student who just picked one. Well, there was actually 2 correct answers here, right? Here's the female, who is the homozygous recessive, and the male is either big B, or a little b, sorry, big B, little b. That's what we're trying to establish in this test cross based on the number of offspring. So if you have any barless offspring, you know he had to carry the recessive allele. If all the offspring were barred, then you know he was homozygous dominant. He had no recessive allele. Okay, so again, students did not read carefully on that. All right, next up was our sex-linked traits. And this information it's very different than autosomal. And a lot of students were treating it as autosomal. That is not correct. So that means that if you are choosing to express your genotypes as big H, little h, that's not right. That shows me you have a basic fundamental misunderstanding. You are not understanding that the autosomes, the body chromosomes, those are body chromosomes number 1 through 22, they are controlling our traits and characteristics like hair color and eye color. Our sex chromosomes, however, that's our 23rd pair of chromosomes. If you are a girl, you have XX chromosomes. If you are a boy, you have XY. And so there are some specific traits that are on these X or sex chromosomes. So here they're telling you straight up that this is X-linked recessive and the female is healthy. That means she has no hemophilia. She does not have this disorder. Yet when she's mated with some healthy males also who do not have the disorder, she has some little pups, her offspring, that do have hemophilia. So because she is normal and she has the normal dominant healthy allele, that must mean then that she carries the recessive, and she has passed this on to some of her male dogs, and they in turn get hemophilia. So she herself is normal. She carries, though, the recessive and passes it on to some of her offspring. So she's bred, mated to one of her males that does not have hemophilia. So he's a normal male, not a hemophilia male, but a normal male. Okay, and so again, if students are choosing big H, little h, that is a fundamental misunderstanding. You have to know that sex-linked is on the X chromosome, sex-linked, X-linked. And then to determine the probability of her females, well, remember, she is a carrier. So could she pass this on to her offspring? Absolutely she could. But when you do the Punnett square, neither of her daughters have hemophilia. They would need an X little h from their dad, and it says right here, he does not have hemophilia, so he is the normal. So there is no way that her kids, her female, puppies, are going to have hemophilia. Zero chance. Okay, so zero can be an answer. Don't be freaked out that you get zero for an answer in genetics. Okay, and again, just a reminder, careful reading. Okay, another couple of examples. Please watch my instructional videos. I have dihybrids and pedigrees, and very important that you follow my instructional videos to learn how to do dihybrids, to find out the correct gametes, and when you're doing a pedigree, to determine the mode of inheritance and to assign genotypes. So I'm not going to have time to go through the cell division summative. If you have any particular questions on it, you may go ahead and ask me. But many students, most of the class actually got this one wrong. And again, it was a reading error. I asked you to begin with the stage in which the nucleus and cell contents divide, and that's mitosis. So number 2 should have been the first in your answer. And again, many students did not read this carefully and did not get the correct answer. Again, if you have any questions, don't hesitate to email me, michelle.ladine@ghsd75.ca. Talk later.